Great. Well, it's very nice to be here today. I'm going to talk about the chemicals that, that plants make, because plants are a rich source of diverse chemistry, as I'm sure you know. And these are some examples of important molecules that plants make. So we have artemisinin up at the top left, which is an anti-malaria compound produced by wormwood. We have Taxol over at the right, which is an anti-cancer drug. Etoposide from mayapple, also an anti-cancer drug. And this very complex molecule here, which I'll come back to later in my talk, which is called QS21. And it's produced by a tree that grows in a certain part of Chile. And it's important because it's an immunostimulant and it goes into vaccines. So these are just a few examples of the complex chemistry that plants are capable of doing. Around 50% of the drugs in current use are either natural products or natural product inspired. And this is likely to be because these molecules have been honed by evolution to do useful things. They're helping plants to survive in their environments through influencing interactions with pests and pathogens and with abiotic stresses. So these molecules have been honed, refined to interact with biological pathways and processes. So they're a rich source of bioactives. The vast majority of these molecules are too complicated to be synthesized by chemistry, at least at commercial scale, with the exception of relatively simple molecules such as aspirin, which we have on the left. So even morphine, which of course is widely used, is not commercially made by chemical synthesis in the laboratory, but still by extraction from the producing plant. And as we learn more about plant genome sequences, it's becoming clear that there's a huge amount of chemistry locked away in there that we haven't yet even begun to think about. So there are over 400,000 species of higher plants on the planet, and this is a massive resource potentially for finding the genes and enzymes for new pathways and chemistries. We currently only have genome sequences for somewhere in the region of, changes every day, but 3,000 to 5,000 plant species. And those sequences are of varying quality. But nevertheless, we can see from our predictions of gene function, although we might not know the precise functions of the genes that we find, that they're likely to be doing things involved in the biosynthesis of specialized metabolites. So I became interested in the triterpenes many years ago for historical reasons, which I'll explain in a minute. And the triterpenes are one of the largest and most structurally complex groups of plant natural products. And you can see some examples here. So in the middle is an antifungal molecule called adenosine, which is produced by oat, and it's antifungal. And then we have glycyrrhizin, licorice, which is anti-inflammatory. We have anti-tumor compounds produced by the cucurbitaceae. We have the health beneficial compounds from ginseng, and then our friend over on the right, this very complicated immunostimulant, QS21. These molecules are all triterpenes. They have triterpene scaffolds with somewhat different formats, and those scaffolds are decorated, they're oxygenated, they have sugars, they have acyl groups, and so on. So these are just a few examples of triterpenes. Why did I get interested in triterpenes? Well, my background is in plant microbe interactions, understanding the mechanistic basis of plant disease. And many years ago, I became interested in a fungal pathogen of wheat, which causes a disease known as take-all disease. And it's called take-all because it literally takes the yield away. This is caused by a soil-borne fungus called Gomminomyces graminis tritici. So this fungus infects the roots, and then it goes up through the vascular tissue and causes substantial effects on yield. There is no effective means of control of this disease. It can cause routinely 5 to 20% yield losses with complete failure of the crop under severe disease conditions. And even worse, the disease severity increases with successive wheat cropping up to a period of, say, 3 years. So you have to take a hit each time you grow wheat, but then the soil responds and starts to control the pathogen. So it's a major problem, no real control mechanism. But back in the 1950s, a lady called Elizabeth Turner at Oxford University was interested in oats. I should say this molecule of adenosine that I showed you in the middle of the earlier slide is only produced by oats. And it turns out that it's produced in the root tips and in the lateral root initials. And we know this because it fluoresces bright blue. So Elizabeth Turner reported the presence of this fluorescent root tip glycoside in oat roots in a paper which she published in 1960. And she speculated that this molecule may be important in protecting oat roots against attack by soil-borne pathogens such as the take-all fungus. And then a number of years later, the Crombies at Nottingham determined the structure of this molecule, which is actually a set of 4 related molecules, but the major one is adenosine A1. And adenosine A1, as I said, is UV fluorescent. It confers strong fluorescence on the tips of oat roots, which you can see in the panel on the top right. There is a wild-type seedling on the left, and the roots are fluorescing bright blue. So we did a chemical mutagenesis many years ago, and we simply screened for seedlings that lost the fluorescence in the roots, and you can see on the right of the top panel, there's one there, just to ask the question whether or not this molecule might be involved in disease resistance. And we generated over 100 mutants in this pathway. At the time that we did this, almost nothing was known about genes and enzymes for triterpene biosynthesis in any plant species. This was way back in the 90s. Nevertheless, we generated mutants, we tested them, and we showed that the mutants did indeed have increased susceptibility to the take-all pathogen, which would normally not infect oats, it infects wheat. But here on the right at the bottom, you can see brown lesions on the roots because these seedlings are now susceptible. So we demonstrated that Elizabeth Turner's prediction was right, that this molecule is protecting oat roots against attack by pathogens. And just to give you some context, the first Arabidopsis genome sequence, the first plant genome sequence was published in December 2000. So this was published in 1999, so you have to get back in the zone. And this is also a time when you made your own back libraries, which we won't go into. But so we would have stopped at the point where we'd shown that these mutants were compromised in disease resistance, because this was a very complex pathway. We expected to be a multi-step pathway. Nothing was known about triterpene biosynthesis. Diploid oats were not a model plant. However, we found that the loci that we'd hit by mutation that we hadn't characterized were not segregating away from each other. So this is classical genetics. And this was suggesting that these genes were organized in a multi-gene locus. This is a snapshot from our early work where we did make a back library and we started to clone the genes. The first 5 genes are shown there. They all encode different types of enzymes in the pathway. They're expressed in the roots. The fluorescence, if you take a cross-section of the root, is in the epidermal cell layer. And you can see that the genes that we've characterized are also expressed in the epidermal cell layer of the root. So the whole pathway is taking part in that cell layer. And then we can skip ahead to 2021 after a huge amount of work, where my friend Bin Han in China very kindly sequenced our diploid oat accession that we've done the mutagenesis on. So this is a pseudochromosome level assembly. And the bottom right, there is a panel showing DNA fish, and the green spots are the gene for the first enzyme in the pathway, which you can see is very close to the end of chromosome 1. And we also completed the characterization of the entire pathway, and you can see that it turns out to be a gene cluster, a biosynthetic gene cluster, as we had predicted from genetics. And I should emphasize that this is not a tandem array. These genes all encode different types of enzymes for the entire pathway. The cluster is close to the end of the chromosome, as I mentioned. The genes within the cluster are all expressed strongly in the root tip with very little expression elsewhere. If you look at the flanking genes, these genes are not part of the pathway and they have a different expression. And very, very strangely, the genes are in approximately the same order as the biochemical pathway, and we can come back to that later. I should emphasize there is no evidence to suggest that this gene cluster has come from microbes. It absolutely hasn't. We can go into that in depth. This cluster has assembled relatively recently in evolutionary time by gene duplication and neofunctionalization. And somehow the formation of a cluster must have been driven by selection pressure. As an aside, we now have a project where we've been working on this for a number of years to try to engineer this pathway into wheat to see if we can generate disease-resistant wheat. Quite how we deploy GM wheat with an entire new pathway, and it is a whole other separate discussion. Interestingly, although the adenosine pathway is particular to oat, and you can see that the fluorescence is in the root tips and the lateral root initials, if we take the promoters from the adenosine pathway and hook them up to a reporter gene such as GUS and put them into different plant species, here it's wheat, but we've also done Arabidopsis, rice, legumes, maize, fern, we see a very similar expression pattern. We see expression in the root tips and in the lateral root initials. So that suggests that this specialized metabolic pathway, which is particular to oat, has somehow plugged into some sort of ancient, highly conserved root regulatory mechanism, which is also very interesting. At the time we discovered this gene cluster, there had been one other example of a biosynthetic gene cluster from plants for a natural product pathway, and that was the work of Alphonse Giel and Monica Frey in Munich. And they had found a gene cluster in maize for the synthesis of a different class of defense compounds, hydroxamic acids, also known as DIMBOA and DIBOA. So the maize gene cluster originally published is shown there, and that pathway is also involved in defense. So then a number of other examples emerged. We went into Arabidopsis to see if we could predict and validate gene clusters there, which we did. Other reports were coming through. This is from a review we published in 2014. And these are examples of the chemistry. So we have our oat compound here. The maize compound is at the top. We found several triterpene pathways in Arabidopsis, which had evolved independently of the oat pathway. There are cyanogenic glycoside pathways in sorghum, lotus, also cassava, diterpene pathways in rice, steroidal glycoalkaloids in tomato, and also alkaloids in poppy. So this is all 2014. This is from a review a few years later where the list is continuing to grow and the chemistry is becoming more diverse. So I'm not telling you that all pathways for natural products are clustered in plant genomes. There are certainly examples where that is not the case. Nevertheless, there are now over 50 examples of biosynthetic gene clusters from plants. And you have to remember that actually we probably only know about 80 or so biosynthetic gene clusters in total in terms of the characterized genes and the genome sequences. So there's a huge amount we don't know. We could speculate for a long time about why some pathways are clustered and others are not. There is no evidence for any of the examples I've shown you so far of horizontal gene transfer from microbes. So these clusters do appear to be forming from plant components. The clusters are conferring a selective advantage, we believe. That's what specialized metabolites do. And so by assembling a new pathway that makes beneficial chemistry, there may be selection pressure to drive cluster formation to enable co-inheritance of beneficial combinations of alleles. And clustering also provides the opportunity for higher level regulation at the level of chromatin and nuclear organization, and there is evidence for that as well. And those two explanations, co-inheritance and regulation, are not mutually exclusive. So it's a very complex area, a lot we could talk about. But there are practicalities. Of course, the phenomenon of clustering enables us to take the kinds of algorithms that have been deployed in microbes to look for biosynthetic gene clusters for new antibiotics and other chemicals and adapt these for use in plants. And so we've collaborated with Marnix Medema at the University of Wageningen, who's developed a number of algorithms for microbial genome mining. And one of them is called anti-SMASH, which some of you may have heard of, and that's for use in bacteria. And so with Marnix, we developed the first customized algorithm for mining plant genomes for biosynthetic gene clusters, which we call Planti-SMASH. Version 2 is about to come out. So this is very early on. It's a broad brushstroke approach. This is an example output for a plant genome sequence. So it spits out predicted biosynthetic gene clusters and just gives you a snapshot of what might be there. These are not all going to be real clusters. They need to be checked and validated, but it's a very useful first start. Like many other groups around the world working on plant natural products, we do use microbes as heterologous expression systems. but we also really like Nicotiana benthamiana, which I'm sure many of you are familiar with. And so we use a method developed by George Lomonosov, my colleague at the John Innes Centre, which involves using a particular set of vectors to do agrobacterium-mediated transient expression. And George developed these vectors initially for protein expression, as in structural proteins, and we started to use it to look at metabolic pathways. It's very quick, so you put your genes of interest into an expression construct, into agrobacterium, introduce the agrobacterium into the leaves. 5 days later, you can see your result. In this case, we've made a fluorescent compound, and also you can see GFP. So it's very quick. It can be scaled up because if you're doing a syringe without a needle infiltration, you're just infiltrating a small circle. But if we want to generate more material, this is just at lab scale. Our engineering department designed a vacuum infiltration system where we can take whole plants and infiltrate them with agrobacterium. And here you can see the leaves from one plant that has been agro-infiltrated. And this is with a GFP expression construct. And you can see bright GFP fluorescence. The smaller leaves at the top are not fluorescing because they grew after the infiltration process had been done. And on the right-hand side, you can see Michael Stevenson, a natural product chemist who now has his own position at the University of East Anglia, also in Norwich. This is his proud face, not his happy face, because he's just extracted a gram of 98% pure triterpene by extracting from a few hundred plants that have been agro-infiltrated. But that's quite something at academic scale to make a gram of 98% pure compound. So this is all very timely because the number of plant genome sequences is growing very rapidly. The transient plant expression system is very quick. As I say, 5 days from doing the infiltration to finding out what you've made. And we can scale up to generate material for NMR to validate structures and also to do bioactivity analysis. So as I said, we've been focusing on the triterpenes, and I gave you some specific examples earlier on. But the really nice thing about triterpenes, there are several very nice things about them. They all come from 2,3-oxidosqualene. 2,3-oxidosqualene is a precursor for the biosynthesis of essential sterols. So it's present in us, it's present in fungi, it's present in Nicotiana benthamiana. That molecule is normally cyclized to sterols as part of housekeeping, essential housekeeping metabolism. However, in plants, it can be cyclized to over 200 different cyclic hydrocarbon scaffolds, of which you can see a few here. So this is one substrate, a big enzyme family, can do a lot of fancy origami and make a lot of structural diversity just at that level. And then those scaffolds can be oxygenated, selectively oxygenated, which you can't do with conventional chemistry, to put handles on that can then be further modified by the addition of sugars and acyl groups and other things. So enormous structural diversity, one starting point, tens of thousands of molecules. It includes the steroids, which are also cyclization products of 2,3-oxidosqualene. And overall, this superfamily of compounds is a rich source of pharmaceutically relevant molecules. So one of the things we did early on, this is work from James Reed, who was doing his PhD in my lab, was we did combinatorial biosynthesis, where we took a triterpene synthase that made the scaffold in the middle. It happens to be beta-amarin. We took 5 cytochrome P450s, which we knew selectively oxygenated that scaffold at different positions. And then we co-expressed each of those P450s with the triterpene synthase. And you can do this simply by mixing the agrobacterium strains that contain the different expression constructs. You don't even have to make a multi-gene construct with all of your genes on it. And that becomes very relevant later on. So James did this with individual P450s, then with pairs of P450s. These genes are from different plant species. So with these 6 starting genes, he was able to make over 40 different selectively oxygenated beta-amarin names. And this couldn't be done with chemistry in the laboratory because you don't have the selectivity. Some of those molecules have previously been reported. Some of them were new to nature because, as I say, he was mixing genes from different plant species. He was able to purify a suite of analogues And with a collaborator over at UEA, Maria O'Connell in the School of Pharmacy, James tested the effects of these molecules on human cell lines and also looked at TNF-alpha release activity. So this is just a proof of concept to show we can make chemistry and we can test it. We then took our pathway from oat, the 12-gene pathway, which is normally expressed in the roots of a monocot, and we showed we could reconstitute it again by transient expression in the leaves of benth, which of course is a dicot. And we haven't had to worry about cofactors or lots of other things that you would need to worry about if you were using microbial systems. So it's very flexible. We then got a bit too carried away because we thought, having done that, we could consider this molecule, which is far more complicated than the oat molecule. So this is QS21, the immunostimulant that I mentioned. It's produced by this tree, which grows in a certain part of Chile. It was approved for use in human vaccines in 2017. And the first one was the shingles vaccine, Shingrix, also malaria, COVID, a respiratory virus vaccine, with a whole load of other vaccines in the pipeline. And it has been valued at between $100,000 and $400,000 a gram. That's debatable, but that's what we've read. So there is a huge amount of interest in this molecule because it's in great demand. And so here you can see the difference between the oat molecule and QS21. They've got the same scaffold in black, some oxygenations, but there are a lot more exotic sugars on QS21. And there's a C18 acyl chain, the origin of which was not known. And that modification is particular to this tree, Kiaha. So that molecule has only been reported from Kiaha. So we started with the 1KP project, which some of you may know. And this is a transcriptome resource from different plant species. There was some Kiaha information in there. We had a rough idea, because of our experience of working with triterpenes, of what we were looking for the first few steps. We knew what a beta-amarin synthase looked like, because these are within the triterpene synthases, they can actually be readily identified based on phylogenetics, unlike most of the other triterpene synthases. And we also found several P450s that converted beta-amarin to queleic acid, which is the oxygenated scaffold of QS21. So the first two steps were basically predicted, inferred. The last one was more challenging, and that was identified by a process of triaging the CYPs that we found and looking for those that were more likely to be involved in specialized metabolism and testing 17 of them. But it's a long way to go from queleic acid to QS21, and the 1KP project has a lot of limitations, including the quality of the assembly of the transcriptome. So we got the tree, and this tree was sourced. I mean, we're free to work with it. It has been in the UK in a nursery for decades, well before the UK signed up to the Nagoya Protocol, and its provenance is well known. It came from Europe before then. And we sequenced the genome of the tree, and you can also see the chromosomes there. So we got a very high quality genome assembly and very good transcriptome resources for different tissues. And then we started to use the 4 genes we characterized to go fishing for the downstream pathway genes using a combination of approaches. These included co-expression analysis. We also used the Planty-Smash approach to look for any candidate biosynthetic gene clusters. And we started out by looking for the genes that added the sugars on at the left-hand side of the molecule, because again, from our knowledge of triterpenes and the structures that have been reported from nature, we thought that this was the most likely starting point. And so we prioritized UDP-dependent sugar transferases. It was a bit more complicated than that, but I can go into that later. And we found the enzymes that add the trisaccharide chain onto the left-hand side of this molecule. There are actually 2 different variants with different sugars, and we got both of those. So that was a step forward. We could use the large-scale vacuum infiltration to purify these, so we could determine their structures by NMR. So again, just flagging the benefits of this system, very quick for analytical, and it can be scaled up to enable preparative scale production of compounds. And after a huge amount of work, we also got the enzymes to add the sugars on at the right-hand side, and Anna El Demirdash, who was a chemist in the lab at the time, purified all of these intermediates and determined all of their structures by comprehensive NMR, which was a massive undertaking. So this is not only revealing the genes and enzymes for the pathways, but also validating the biosynthetic intermediates. And then we came to the addition of this acyl chain. We didn't know where it came from. We made some predictions. There's also a sugar attached to it. And then by this point, our computational platform had become very advanced, and we were factoring in not only co-expression, but also levels of expression in particular tissue types, presence in a predicted biosynthetic gene cluster, presence in a cluster that contained at least one gene which shown was in the pathway. This enabled us to take the 30,000 or so genes from the whole of the genome and to devise a scoring system where we prioritize the top 50 or so. And when we do this, the ones in grey are the ones we've already characterized, and we were looking for the missing steps to add the acyl chain and the final sugar. And those candidate genes are shown in different colors here. And then Letitia Martin, a postdoc in the lab, decided, so we already had all of the genes to make the advanced intermediate that you can see at the top. We'd worked out how the acyl chain was made, which I won't go into, but there are 2 separate C9 acyl units that are added. We had the genes for those. She threw all of those into the mix with all of the candidate genes and co-infiltrated around 70 genes in one go, which I said would never work. But she got this little peak that had the right mass, and it turned out it was QS21. So 70-something genes co-expressed. I mean, she deconvoluted all of that. We worked out what the steps were. So we've basically sequenced the genome of the tree, elucidated the pathways not only to QS21, but also to a simpler molecule called QS7, which is another saponin. The tree makes over 100 structurally related molecules, including QS21 and QS7. QS21 is used in vaccines because it's an immunostimulant and it's relatively abundant in bark extract, but it's also quite toxic to humans and has to be carefully formulated. QS7 is a very good immunostimulant, is less toxic, but is present in vanishingly small amounts in bark extract. But now we have the instruction manual to start to make QS21, QS7 and variants of these and systematically explore the relationship between structure activity and adjuvant properties. So just to start to wrap up, the amount of plant genome sequence is rapidly growing. This is now, of course, out of date. I need to update this slide. And there are these major sequencing initiatives ongoing to sequence every species on the planet, the Earth Biogenome Project, for example, and in the UK, the Darwin Tree of Life Project. And these genomes contain the instruction manual to make a huge array of chemistry, most of which we have not yet discovered or even imagined. We work with lots of different species, not just oat and kiaha, and we just go where our interests take us. So we've worked on defense-related molecules in wheat. We've worked on the Brassicaceae. Soapwort is very interesting. That's a member of the Caryophyllaceae. It's very distinct from the Chilean tree, which is a Phobales. But soapwort actually goes quite a long way towards making advanced triterpene scaffolds that look like the QS21 pathway. So that is very interesting. We've elucidated the whole of that pathway and again reconstituted it in Benth. I should say that the Kiaha work took 6 postdocs several years. The soapwort work took one student, Jenny Jo, 3 years to elucidate the whole pathway. But I should point out that she didn't just find the homologs from Kiaha, because Kiaha and soapwort are taxonomically remote. She had to do all of that de novo. But because our platforms have advanced so much, it's now becoming much quicker to go through this loop. We're working on anti-insect compounds from Chinaberry, which is a relative of neem. We've discovered over 50 previously unreported triterpenes in Arabidopsis that have roles in shaping the root microbiome. So this is in the gold standard model that we thought everybody understood. We've also gone into sea cucumber, which is one of the few animals that makes defense-related triterpenes, but it didn't stop me eating that bowl of sea cucumber soup that I was given in China. So as well as looking at individual pathways, we can start to take much bigger approaches. I've mentioned mixing and matching enzymes from across the plant kingdom. We now have a toolkit of hundreds of enzymes to make and modify triterpene scaffolds. Where there are sufficient genome resources, we can look across the genome sequences of related species in the Brassicaceae or in Arabidopsis and start to infer based on associations of flavors of genes within gene clusters, for example. And we're working on enzyme superfamilies using machine learning-based approaches with Janet Thornton and Christina Rengo to try to predict the chemistry from the gene sequence. We've inadvertently made a whole load of compounds. We've made thousands of compounds during the work that we've been doing. We went through the freezers to find out how much we had of purified compounds to just do an initial test with OncoLead, a company in Germany that tests compounds against cancer cell lines. So we sent a library of 32 or so compounds to OncoLead. And this is just, we're just looking to see what kinds of bioactivity is there. So this is just the beginnings of starting to get more of a meta view of the properties of these molecules. So I'd just like to finish then by saying that in the 19th century, advances in chemistry led to the beginning of the pharmaceutical industry. And now we're very much into the bio-revolution, where we can take the instructions from plants and we can go through a loop where we work out what these genes do. We can discover new genes, enzymes, pathways, and chemistries. We can scale up as needed. We can do bioactivity assays. Importantly, we can use machine learning to not only try to predict chemistry from DNA, but we can learn the chemical properties of molecules from the public databases and from in-house databases to understand what features of molecules are associated with particular bioactivities for particular indications. And in that way, we can inform the way in which we go around this loop with the ultimate aim of harnessing the chemical engineering capability of the plant kingdom to make designer molecules for particular applications. So I would like to finish there just by drawing your attention to the many people who've contributed to this work, including our collaborators and also our funding sources.